So let's continue our basic description of these two large domains, bacteria and archaea. As we talk, I want you to keep in mind that although the bacteria and the archaea have a lot in common, they are two separate domains for a reason. It turns out that the archaea in many cases are more like the eukaryotes than they are the bacteria. So as we talk about this, even though they do share a lot in common, there are some major differences, and I'll try to highlight those as well. Let's talk a little bit about the internal organization and the DNA of these creatures. Both the archaea and the bacteria are prokaryotic. We've already talked about that, meaning they usually lack most of the organelles associated with eukaryotes, and they lack the complex compartmentalization. They don't have things like mitochondria and endoplasmic reticulum and all the various compartments and pieces that a eukaryotic cells usually have. Some prokaryotes do have some specialized membranes that perform metabolic functions, usually various kind of folded portions, infoldings of the plasma membrane. Some aerobic prokaryotes have infoldings similar to the cristae of mitochondria. So in certain aerobic prokaryotes you can find portions of their cell that sort of look like or remind you of the mitochondria, and some photosynthetic prokaryotes, particularly one called cyanobacteria, have thylakoid membranes much like those found in chloroplasts. And so we're going to talk a lot about endosymbiosis, which was the idea that one cell engulfed another, and that's how we developed into eukaryotic cells, that we'll talk about later. Well, this is a good kind of clue or kind of a lead into this. So the mitochondria of a eukaryotic cell looks very similar to certain kinds of aerobic prokaryotes, and the chloroplast of a plant cell looks very similar to a cyanobacteria. And so we're going to discuss that further in just a moment. So here's an aerobic prokaryote that has certain infoldings called respiratory membranes that looks something similar to what you might find the cristae of a mitochondria. And this photosynthetic prokaryote has thylakoid membranes and is very similar and looks quite a bit like the chloroplast, say, of a eukaryotic plant cell. If we look at the DNA of a prokaryotic creature, for the most part, prokaryotic genome is smaller, has less DNA than a eukaryotic genome, and most of the genome consists of a single circular chromosome, as opposed to eukaryotic cells, which generally have multiple linear chromosomes. So the prokaryotic has usually a single circular chromosome. Sometimes they also have some smaller pieces of DNA called plasmids, which I'll show you a picture of. The chromosome is not going to be located inside its own membrane. It doesn't have a nucleus, but it is usually located in something called the nucleoid region of the cell. And again, like I mentioned, some species of bacteria have the regular genome, the regular circular chromosome, which is fairly large sometimes, but then they also have smaller separate little rings of DNA called plasmids. In this picture, this is an E. coli, probably the most famous or well-known bacteria that we've studied more than any other, and it has ruptured, and so it has released its singular circular chromosome, and so you can see all these little lines. That's part of its DNA, its chromosome. And then kind of off to the side you can see some little plasmids, which are separate circular DNA pieces right here. So excellent example, kind of a very cool photograph taken from a very high powered microscope. Even though DNA is in all living creatures, and DNA is the same in all living creatures as far as being made up of the four nucleic acids that we've already discussed, there are some differences between prokaryotes and eukaryotes in DNA replication, transcription, translocation. For example, the prokaryotic ribosomes are slightly smaller and they differ in their protein and RNA content. So there are some differences, but the basic feature of DNA, for example, is the same in all living creatures. These differences allow people to use some antibiotics, again, such as tetracycline, to inhibit bacterial growth without harming themselves. So that's a very good benefit for humans or for our pets or whoever else we might give antibiotics to, because these medications will attack the differences between the prokaryotic and the eukaryotic cell. They won't negatively affect the eukaryotic cells, but they will negatively affect the prokaryotic. So you can take that antibiotic and it won't harm you, but it will potentially, hopefully, kill off the bacteria. As you can likely imagine, reproduction and adaptation is quite a bit different in a unicellular prokaryote as opposed to, say, a multicellular animal or plant. Prokaryotes can produce pretty darn quickly through binary fission, where they simply divide in two and they create two daughter clone cells of each other. They can divide usually about every 1 to 3 hours on average, depending on conditions. In some cases, they can go as fast as every 20 minutes. If they have all the nutrients and perfect environmental qualifications, say one E. coli bacteria could divide undergo binary fission every 20 minutes. If it were to do that unchecked, if every 20 minutes that bacteria divided and then those two became 4 and those 4 became 8, you would have, within 2 days, within 48 hours, you would have a colony of E. coli bacteria that would outweigh the entire planet Earth. So obviously that doesn't happen because environmental conditions keep that in check. But if the E. coli had everything it needed and it divided every 20 minutes, you would have a colony of bacteria that outweighed the planet in about 48 hours. This ability to rapidly reproduce when it has the optimal environment is really a big factor in their evolutionary success. One cool thing to keep in mind is you have all sorts of bacteria within your own body. If you were to count up the number of bacteria cells and the number of human body cells, again, we're talking about the number, and keep in mind bacterial cells are quite a bit smaller, but if you're just going by the number, there are 10 times as many bacterial cells in your body than human cells. There's even a link here to a very cool little article that talks about that. So that means when you think of you, who you are when you look in the mirror, you are more bacteria than you are human if you're counting the number of cells. Now, of course, these bacterial cells can't constantly reproduce because there are environmental pressures that kind of keep them in check. Same thing that keeps the number of lions in check or the number of grasshoppers in check. It's going to be the lack of nutrients, the buildup of waste products, competition, predation by other organisms, that type of thing. So the key features in the ability of the prokaryotes to reproduce is the fact that they're very small, they reproduce very quickly by binary fission, and they have very short generation times. So when things are good, they can reproduce pretty wildly and they can do that very quickly. But what about when conditions are poor, when they don't have enough nutrients or the temperature isn't right? Well, they have an amazing ability, prokaryotes do, to form what we call metabolically inactive endospores. And think of an endospore kind of as a seed, a bacteria seed, and that seed, that endospore, can remain viable in pretty harsh conditions for a long time, and in some cases hundreds of years, centuries. What happens is the cell produces a copy of its chromosome, and around that chromosome it forms this really tough multi-layer shell, and then it removes all the water from that, and again it becomes almost like a seed, like a hard bacterial seed, and then the bacteria, the parent bacteria, will lyse or split open and release that endospore into the environment. And that endospore in many cases can withstand incredible environmental conditions. It can withstand boiling water, lack of water, incorrect temperatures, and it can stay viable but dormant for sometimes centuries at a time. Last thing to keep in mind about these prokaryotes as far as their short generation time and their reproductive cycle is because they have such a short generation time, they can reproduce so quickly that allows them to evolve very quickly. For example, adaptive evolution in a bacterial colony has been documented in the lab over just an 8-year period. There's a very well-known set of experiments that has been going on now for quite some time, probably 8, 10, 12 years now, by Dr. Linsky. If you look on page 561, and you can look on the internet for the various Linsky E. coli experiments, this guy has been studying E. coli, and his lab and his partners have been doing experiments for 8, 10, 12 years, and they've studied over 50,000 generations of E. coli bacteria, and they have seen evolution in action. And you can see that in these E. coli because their generation time is so short. I mean, the E. coli can reproduce, every 20 minutes or every 45 minutes, whereas it takes a human, 15, 16, 20 years to reproduce. So that short generation time allows you to kind of see evolution in your own lifetime. And the last thing I want to remember is that these prokaryotes, we talk about them as if they're simple, primitive creatures, but you have to keep in mind they have been evolving for 3.5 billion years, so they are not really primitive. They are very highly evolved, and they're very well suited for their environment, and that evolution over that huge amount of time has allowed them to be successful in extremely variable environments, and they outnumber us, and they can live in places where we can't live. So they are well evolved and not exactly primitive. Last little slide before we take a break here. This is part of the Linsky experiments that I was talking about, where it shows how rapidly these creatures can evolve. These prokaryotes can evolve in response to an environmental change simply because their generation time is so fast. You can build up changes, environmental or evolutionary changes, through mutations and natural selection in a very short amount of time. And this one experiment they talk about in your textbook kind of shows that. We're going to take a break right here. We'll come back and talk more about these prokaryotes in just 1 second.